Okay, so I'll say to everyone on Zoom, since there's the two of us here, we're very excited to welcome Will Towns from the Department of Computer Science at Princeton University, and he'll be talking to us all about spatial factorizations.
Thank you. And thanks to everyone for such a fantastic visit. I had so much fun getting to meet everybody. So before I get into the research part of the talk, I just wanted to briefly mention actually, is the Zoom stuff showing on people's screens? Yeah. Just realized that. Where did it go? Oh, great. Okay. Yeah. So just a little bit about me. I had a very non-traditional background and a different path to biostatistics. I started out doing tropical field ecology, and this is me meeting with a community of indigenous people in the Philippines, trying to get their permission to do research on their land. And this is one of my guides, and this was the plant that I studied. So my first publication is about seed dispersal of this this particular plant called Leah. And another interesting tidbit is my first statistics class I took at a community college. So, whoops, this thing doesn't seem to be.
Just click back on the, like use the mouse to be on the, and then it should be good.
Oh, because it thinks we're still.
Yeah.
Okay, so an overview of my talk. First, I'll introduce the type of data that I've been working with, single-cell RNA-seq and spatial transcriptomics, different ways of measuring gene expression, for those who are not familiar. And I'll talk about my approach, which I call non-negative spatial factorization, and from a modeling standpoint, and compare and contrast with some other possible models that might be of interest for this type of data. Then I'll go into a bit more detail about the actual inference strategy that I took, which is variational inference, and some of the tie-ins to other Gaussian process types of models, and a brief mention some topics that I'm interested in for the future and some of my other projects that I've worked on over the years. So this is an illustration with fruit of how different types of measurement strategies for gene expression could be characterized. So if you think of each piece of fruit as a single cell, and the tissue context is this tart where you have different cell types arranged in a very spatially characterized pattern. That's how a lot of real biological tissues are. But historically, the only way that we could measure the activity of the cells was to kind of grind them all up into a slurry and average across all of them in bulk RNA-seq or in microarrays. More recently, single-cell RNA-seq was developed to allow us to profile gene expression in individual cells. However, in order to do this, we had to dissociate them from their spatial context. And with spatial transcriptomics, we're able to actually retain that spatial context. So here's a kind of cartoon illustration of the way that the molecules are actually captured. It's much like an urn model. The original cell or sample has a large number of molecules of messenger RNA, and we take a sort of random sample of those through some complicated biochemical process. What we actually observe is a small subset of the actual molecules in the sample. So we might be interested in estimating the true proportions of different RNA species or different gene activity levels given this small discrete sample. This is more details about the complicated biochemistry. Particular interest is the fact that with single-cell RNA-seq and also spatial transcriptomics, the amount of RNA starting material is very low. So you have to do many rounds of amplification to increase it so that we can pass it to a sequencing machine. And that can introduce a lot of biases and distortions in the data, the more cycles of PCR that you do. So that's definitely a big consideration. And in case you're interested to see what the data actually looks like, this is a particular data set from Decomposition et al. The breast cancer data, triple negative breast cancer. And each column here is a cell, a single cell, and the rows are representing various genes. This is not the full data matrix. This is a tiny subset just for visualization purposes. And one thing you can notice already is some cells have higher expression levels across all genes, and some genes have high expression levels across all cells, but it's not necessarily uniform. So there's some questions about normalization and standardization that come into play. And furthermore, a large majority of the counts are actually zero. Spatial transcriptomics, we're taking a thin slice of tissue and laying it down on a plate. And the plate has a tiling of probes. Each probe has a barcode to indicate the unique spatial location, the X and Y coordinates. And that's actually encoded into a molecule. And that molecule will stick to the ends of the messenger RNA and bind them so that after a lot of biochemistry, we can then pass that to sequencing and we can actually read out the high dimensional gene expression vector for each of the individual spatial locations. And there are, of course, many other possible techniques. This is a very early technique with a small number of spatial locations. Now we have tens of thousands of spatial locations, and it's increasing all the time. So this type of data schematically might be represented as a high dimensional count outcome, which is also found with single cell RNA-seq. But now we have associated with each observation its spatial coordinates. And typically this is a two-dimensional vector. Of course, in the future, we hope that we can have 3 dimensions or even 4 if we have spatio-temporal techniques. In terms of the order of magnitude, we're thinking of hundreds to tens of thousands of observations, and the number of genes is on the order of a few thousand to say 20,000. But in many of these types of data, we find that many genes have no information because it's within tissue rather than between tissues. So it's very common to perform a initial feature selection step to reduce from say 20,000 genes down to about 2,000 or 5,000 highly variable genes or spatially variable genes. Here's what the data actually look like for an example from 10x Visium, the anterior sagittal section of a mouse brain. You can see that, again, so here it's transposed from before. I'm moving from R layout to Python. So here each row is an individual spatial location, an individual observation. It's not a single cell because for this particular technology, the spatial resolution is not small enough to capture individual cells. So each observation could be thought of as an average, a local average of say 5 cells, depending on the size of the cell. And we can see again that there's a lot of vertical stripes, meaning that there's certain genes that are highly expressed across nearly all of the locations. But even if you look at the first position here, you can see this gene is not expressed anywhere except for a very small subset of observations. So that gene might be a marker for a particular cell type. So that should give you a flavor of the type of data that we're looking at. Now I'll introduce the modeling approach, and I'm going to build up from simpler models to the actual model that we used and show how they're all related to each other. So what are some goals that we would have for analysis of spatial transcriptomics? First of all, we're very interested in dimension reduction because it's even with 2,000 variable genes, it's a lot of genes to try to interpret. And because genes are well known to interact with other genes, it's very reasonable to assume that that the data actually lie on some kind of low dimensional manifold. And so one way to encode that assumption or that prior knowledge is to think of our high-dimensional outcome vector is some sort of linear or nonlinear combination of a much smaller number of latent functional components, or I sometimes refer to them as principal surfaces. So canonical patterns of variation across space. We might also be interested in interpolation of missing locations, and very important is interpretation. We want to get some kind of exploratory insights into this massive data so that we can guide downstream validation experiments and perturbation experiments and things of that sort. So we might want to, oh, I'm sorry, question?
Oh, yeah, quick question. Just because it was a really clear description to me when you had the plate. And then I'm just trying to understand, because this is not my area, like, when you said missing, is it missing for a reason? Like, is it missing, like, when I think of spatial data missing, is it on purpose? Like you just didn't have pull this particular location, or is it missing because the technology broke? And so it's like missing this more at random, or is it kind of?
Yeah, that's a great question. So I have a figure that might help answer that later, but just quickly, if you look at the tissue slice here, sometimes just the process of slicing a tissue very, very thin, it might get a little wrinkled, or there might be little gaps. You can see holes, here, where it's possible that, I mean, this is a real anatomical structure called a ventricle, where there really is nothing there. It's a tissue, fluid canal. But these holes are actually likely to be just an artifact of the preparation. And so, I mean, it's not the primary focus, but it could be of interest, particularly for future work with aligning multiple slices that you want to fill in the gaps.
Gotcha. Okay, thank you.
So they could be missing. I'm not sure if it's missing at random, but it's sort of like with time series. You want to interpolate where somebody happened to turn their monitor off. And there's various strategies for that. So we might want to identify genes that are not only variable, but actually spatially variable, that the way that they vary across observations is following a spatial pattern. That's something that a lot of folks have been working on. And then of more recent interest for me is trying to determine the relative importance of spatial versus non-spatial variation to different cells or to different genes. So for example, it might not be reasonable to assume that every cell type is strongly correlated in terms of its gene expression with its nearest neighbor cells, because maybe take immune cells, for example, they might be interacting with other cells, or glial cells might be interacting with other cell types. So that's something that we would like to be able to answer. These are some desired properties for an integrated approach to analysis of spatial transcriptomics. We'd like to have a probabilistic model so we can think about uncertainty quantification and prediction. We'd like to have a count likelihood because the data are counts. We'd like to be able to handle gene-gene correlation by assuming a multivariate structure as opposed to individually analyzing one gene at a time. And of course, we want to be able to incorporate our knowledge of the spatial coordinates. We don't want to ignore that. And the last one, we prefer to have a non-negative representation. And I'll show why later, but it basically becomes more interpretable. It produces more sparsity in the decomposition of this high-dimensional matrix, and it tends to identify a parts-based representation. I'll talk about that more in a moment. So one very simple approach to dimension reduction generally is to use principal component analysis, or PCA, which is extremely fast and popular algorithm. And I'm using a somewhat non-standard representation of PCA here. So this is actually probabilistic PCA, but the maximum likelihood estimator for the latent loadings and factors here converges to the traditional PCA solution as you let the variance term go to 0. I'm writing it this way so that you'll see the connection with later probabilistic methods. But it doesn't really check any of our boxes. So this is a method that I developed in the past called generalized principal component analysis, and tried to advocate for its use in single-cell RNA-seq, essentially taking the same low-rank decomposition as principal component analysis, but passing it through a link function, much as you would with a generalized linear model. Unfortunately, this does not produce a non-negative representation. All of these are real valued, and it totally ignores the spatial information, and it's not probabilistic. Factor analysis is a very similar approach as principal component analysis. It is a probabilistic model, and we allow each gene or each feature to have a different variance term, but it still ignores spatial information. Non-negative matrix factorization, or its probabilistic formulation, which I'm showing here, provides many of the desired properties, but it also ignores spatial information. And so the most important thing here is we're again having a kind of inner product. This is in every single model. We have this linear combination, but we constrain the weights or the loadings to be non-negative, and we enforce this non-negativity of the factors through a Bayesian prior. That's what makes it probabilistic. And then also we model this directly as the kind of rate parameter of a Poisson. And this nu I is just a fixed size factor to adjust for differences in the total counts. The standard practice is equivalent to normalization. Okay, moving to spatial models. Spatial DE was invented by Svensson et al. Several years ago. It's a probabilistic model using Gaussian processes, one gene at a time. So it's not multivariate, but it was an initial approach to modeling spatial transcriptomics, decomposing the variation into a spatial component and a non-spatial noise component. This is how it could be represented in a probabilistic way. And they didn't use a count likelihood, they used normalization procedure in order to apply a Gaussian likelihood for computational convenience. So now we're getting into the real stuff of interest. So real valued spatial factorization, this is a term that I'm using. So it goes by many other terms, linear model, coregionalization, Gaussian process factor analysis. But the basic idea is, the initial stages of the hierarchical model are identical to factor analysis. However, rather than using independent priors over the factors, we bring in spatial structure through a Gaussian process. So this is a great approach. However, it doesn't produce non-negative representations. And I'll show you later, it has some limitations. I think I already mentioned this. There are several other models that are more or less equivalent in terms of their probabilistic structure to real valued spatial factorization. A notable one is Mephisto, which was developed by Latent et al. And the thing that's interesting about Mephisto is they apply a kind of spike and slab prior to the weights to try to enforce the sparsity because it's inherently real valued. So you sort of have to force it to become sparse, and that does help with interpretability somewhat. Now, another aspect of Mephisto that differs from our implementation is the choice of kernel function. So in the Gaussian process, so Bayesian, or it's really a probability distribution over functions. And the type of function that can be drawn from this distribution is mostly determined by this kernel function. And so there's many possible choices of kernel, and here I've illustrated several. The two popular families are the squared exponential, or radial basis function kernel, and the class of Matern kernels. So it turns out that the Matern class includes the squared exponential as a limiting case. You let this smoothness parameter go to infinity. This smoothness parameter, this is actually 5/2. It's just hard to get it in the plot. So the smaller the smoothness parameter, going down to 1/2, that's actually equivalent to an exponential kernel, or I believe it's known as an Ornstein-Uhlenbeck in the time series in differential equation literature. is a very rough family of functions. So they're essentially nowhere differentiable. And for our implementation, we chose the matern 3/2. So the idea is that with the biological tissues, such as in the brain, you could definitely see sharp transitions between expression patterns between layers of tissue. So you don't want to have an infinitely smooth function to describe that. It's not going to fit the data very well. Unfortunately, this was the choice. This is typically the default choice with Gaussian processes. And it was used by Mephisto. And we found that it led to worse fit to the data compared to the matern 3/2. So finally, non-negative spatial factorization, which is the title of the talk, checks all of the boxes, provides everything that we want: non-negative representation, spatial awareness through Gaussian processes. But note, much like probabilistic non-negative matrix factorization, we are exponentiating the realization of the Gaussian process function, which means it's sort of a log Gaussian process. And this tiny operation here ruins the convenient properties of Gaussian processes, meaning that we can't have closed form inference. Also the fact that we have a non-conjugate likelihood. So just a small change to the model leads to many challenges when it comes to inference. So here's just a broad overview of all the different models that I've mentioned so far. You can see that our NSF model, non-negative spatial factorization, is right in the middle of all the desired qualities. So in addition to a purely spatial model like NSF, we wanted to be able to incorporate non-spatial factors. So we basically decided that we would allocate, let's say, T of the factors to be spatial ones, and the remaining L minus T to be non-spatial. So this will allow us to assess the relative importance of spatial versus non-spatial variation. Now, here's an illustration of why it's important to have a non-negative part space representation. So we did some simulations with these canonical patterns, which are called the quilt patterns. And we're drawing, I'll try not to go into too many details, but we're drawing something like genes, and each gene is associated with one of these patterns in terms of its spatial pattern. And then we look at, we draw a negative binomial count vector from that latent mean. When we apply the various dimension reduction algorithms or models fit to the simulated data, you can see that the real valued models, while they do actually fit the data very well in terms of deviance or whatever criterion, the interpretation of the latent factors is very jumbled. They sort of combine all of the factors together and average them. And so this phenomenon was noted by Li and Sung in the original non-negative matrix factorization paper, where they applied a non-negative and a real value decomposition to faces. And real valued approaches tended to learn eigenfaces. Each factor was an average of many possible faces. It's a holistic representation. Whereas a non-negative approach would decompose a face into an eye or a nose or an ear, which is somewhat more interpretable. And that's what you see here, non-negative spatial factorization, pulling out separate spatial patterns. Of course, there's some noise in the data that doesn't learn it perfectly, but go ahead.
Yeah, could you tell the difference between RSF and Mephisto? So you said that the spike and slab fire is one thing, and is also the prior is another thing, the exponential versus what are the differences?
Right. So what we found was that sparsity is not that important for this task.
Okay.
It definitely does make gene expression a little more interpretable because you can sort of uniquely identify, which factor each gene is loading onto. But here you can see the result of fitting Mephisto. And the spike and slab is not producing, it's not recovering the true latent space.
I see. So you think it's because of priors?
I think it's because of non-negativity.
Okay.
Yeah. In fact, I don't have this in the slide, but if you simply run scikit learns non-negative matrix factorization on this, you get a result very similar to here. So in this particular example, using the spatial information wasn't the most important thing, it was the non-negativity. So moving on to some real data, here's 3 examples of spatial transcriptomics data that we examined. Widely varying characteristics, some with more than 30,000 observations, and a very, very focused field of view from the hippocampus. Another one with a very wide field of view over a liver model. These are all mouse data. And the Visium sort of intermediate. So there's many possible protocols. So first we did a benchmarking task where we held out a subset of the spatial observations and we tried to randomly disperse across the field of view. And we tried to, predict as a sort of held out prediction task. It's a very common thing to do with a supervised model. What we found was, surprisingly, at least in this data set, real valued models outperform non-negative models, even when they more or less ignored the spatial locations. And this is because there's so much information in the high dimensional outcome. However, within the class of real valued models, you can definitely see a reduction in the error. So the lower deviance is a better goodness of fit. You can see moving from factor analysis, which ignores spatial information, to real valued spatial factorization is a significant reduction in the error. But surprisingly, Mephisto did not perform better than factor analysis. And our belief is that it's due to the choice of kernel function, because otherwise there's very little difference between Mephisto and RSF. Yeah, go ahead.
With RSF, is it just Mephisto where that parameter is allowed to be chosen? Is it data-driven choice, or is RSF move beyond that? I was confused when you said Mephisto compared to RSF. Because Mephisto is a squared exponential.
That's right.
And then RSF is just any choice of the
No, it's the
return?
return 3/2.
Okay, it's return 3/2.
So you can actually try to learn the smoothest parameter, but it involves Bessel functions and differentiating through those things is a huge pain.
Gotcha.
So we just tried some different integer values or half integer values. So that kernel function doesn't have a closed form except for half integer values of the smoothness parameter. So
gotcha. So 3/2 work better than the limiting case.
Exactly.
But it might be that 3/2 compared to a half or 5/2 might be
sure.
slightly,
I can see there is much room for exploration of kernel hyperparameters.
No, I just want to make sure I understand.
Yeah, we didn't spend a lot of time optimizing that because we felt that things like non-negativity based on the simulations were much more important than. But however, I should say we are learning the length scales, the amplitudes, basically all the other kernel hyperparameters. And we're using another small difference. We're using not anisotropic as opposed to isotropic kernels, but that doesn't actually matter that much for the particular data we looked at. So yeah, sure. So again, looking at within the class of non-negative factor models, moving from a non-spatial probabilistic NMF to either our hybrid NSF or the fully spatial NSF also reduce the error on this particular data set. Now here's the same task with a different data set, and you can see the results are quite different. Now we're seeing that there's no particular advantage of using a real value factorization, although the RSF is still the top performer. In terms of goodness of fit or prediction error, we're actually getting excellent performance from our non-negative spatial factorization. So it's hard to make a general claim about the superiority of one model purely from a prediction standpoint. So
I was surprised that the NSF-H is actually performing worse than why is that?
So this is for prediction on held out data. So what we would expect is using that spatial information would be critical for that, because you're trying to learn the expression of a point that's right next to a point where you have an observed training data. So the fact that this non-negative spatial factorization, so let's say it has 36 spatial factors. So every one of those factors is able to leverage spatial information.
Limit the dimensions, so you're basically having reduce the
right
dimensions.
So this one, even though it has 36 dimensions, latent components, only 18 of them are spatial.
Okay.
So the other 18 are just sort of noise, but they're not noise. They're learning stuff from the gene expression alone. Yeah. And here Mephisto actually does the worst. Again, I suspect it's due to this very strong smoothness assumption in the kernel. And, Mephisto, I've contacted them, and I wouldn't be surprised if in very short order they switch to the Matern kernel. So if that happens, then I'll be very happy with with my work, because a lot of people aren't even aware that Matern kernel is a possibility. Now, in terms of computational speed, here we're assessing on time to convergence of these various models on a 30,000 plus observation data set. So obviously, using Gaussian processes, even with our inference, tricks, it's going to be quite a bit slower than a simple approach, that ignores spatial correlation. But we were pleased that our implementation was definitely competitive with the competitor Mephisto implementation.
So why the ISF is also slow?
Yeah, I think I'm not totally sure. So one thing you note is that it increases with the number of latent components, because there's more parameters to learn. I suspect that there could be some numerical difficulties there, because especially with the real valued model, the parameters can go off in all different directions. You can have one that gets really, really big and another one that gets really, really small, and they kind of cancel out. So I think there could be some more work done there to, build in some kind of shrinkage priors or something to stabilize the numerics of it. I think it tends to wander around a lot in a much larger set of parameter values, and it might take longer to reach convergence. It's just a speculation. I don't have strong evidence to back it up. Yeah, in terms of sparsity, Mephisto actually does a great job, because it explicitly encodes sparsity through the prior, but our just simple non-negativity constraint also produces quite a bit of sparsity in our loadings. And we're pleased to see that the sparsity increases with the number of latent components. So this is helping us to avoid overfitting. We can increase the number of components without the risk of learning redundant components. Of course, it's not perfect. There are some redundancies, as you'll see, but for the most part, it seems to be doing the right thing. Now, this is what our factors look like, our latent principal surfaces. And this is the hippocampus, a coronal section of the mouse hippocampus with about 33,000 observations. And the red means that the factor has a high value, and blue means that it's close to 0. It's non-negative. And what's interesting is we were able to pull out, without any prior knowledge put into the model, we can actually segment out this tissue. So here we have, I think this is the core. I had to learn a lot of neuroanatomy. This is the choroid plexus. This is the medial habenula. This is the thalamus. This is part of the hippocampus. It's called the Ammon's horn. Cerebral cortex, dentate gyrus, corpus callosum, fiber tracts, and this is my favorite, number 10. Apologies to those of you on Zoom. I hope you're able to see what I was talking about. But number 10 is actually, it was a mystery to us. We thought it was an artifact, or I showed it to a neuroscience friend and they said, it doesn't look right, something's not right. So I went in and looked at the loadings and tried to examine the genes that were heavily loaded onto this factor. And it turned out it's real. It's the meninges. It's a very thin wrapper layer around the brain. And I was very excited that we were able to detect that in an unsupervised way.
So this is really impressive reconstruction of different cell types. So this is a really good reconstruction. Do you have you done this kind of thing for other methods, and how they compare, like in terms of how, is it something that other methods could pick up like this precisely, or have you
that's a good question. So I can tell you that Mephisto, we had trouble applying Mephisto to this data. This is the biggest data set we tried. It tended to run out of memory, so I wasn't able to make this plot with Mephisto. But in the original Mephisto paper, if you check it out, they do have, maybe I'll show you the next, let me come back to that if you don't mind, because I have another data set that they did compare against, and they did learn some of the similar patterns. I think it would struggle to find these very, very thin spatially localized areas, particularly with the choice of kernel and the lack of non-negativity. You can make a lot of progress actually with non-negative matrix factorization. There, the difficulty is you don't learn functions. You're just learning a vector for each of your observed locations. Trying to generalize that to a whole surface is not totally straightforward.
What I'm just curious about is, what's the, how does it become when you don't apply Gaussian process? How it
Right.
become when you don't do the non-negative factorization, which is all, I think really good combination. It's a computationally challenging, but yeah, it's a very interesting result.
Yeah, thanks. I really enjoy looking at that figure. I like I had to fiddle around with a lot of color schemes in Matplotlib. But here what I wanted to show you is that for each of these factors, we can pick out the corresponding gene or genes that are most associated with that based on our non-negative loadings matrix. And so then we can just plot the expression levels of that gene across space, and we can see that it more or less corresponds to the same pattern that we observe in the factors. The factors are much more crisply defined. If you think about this number 2, the thalamus, it's very clear where the boundaries are. But if you were to look at the top marker gene for thalamus, it's quite a bit noisier. So the advantage of this multivariate approach is we're able to leverage many genes that are correlated with each other to learn this representation, as opposed to going through one gene at a time and looking at plots like this, some of which may be strong, like TTR is a very strong indicator for the choroid plexus. But many of the other tissue regions don't have one gene. It's a combination of genes working together. Now, I'm also plotting here all of the non-spatial factors that we learned from this hybrid model. And we don't really expect to see spatial patterns in the the non-spatial components. Of course, it's not a perfect decomposition. There's nothing stopping these from learning spatial patterns if they want to. It just doesn't have a prior that encourages it. So they're not really anti-spatial. They're just non-spatial. But there's really not much to see here. We were able to identify some of them mapping to particular cell types, but of course they're of less interest in the spatial factors. Now here's another data set with a mouse liver using the XYZ protocol. And here each spatial location includes more than one cell, but we do know the single cell identity. And in the spatial components, we can see a core here of hepatocytes. We learned that from looking at the loadings and the genes that are overexpressed there. And then in the other components, we can see, especially this one, number 3, it's identifying a different region of the tissue, which is actually a tumor that's starting to invade. Now, this data set from Bayesian was also analyzed by Mephisto. And they were able to learn patterns like number 2 and number 3, very broad, patterns. I think they also learned number 10. But I didn't see patterns like number 7, learning some finer grained resolution in the olfactory bulb. So in case you're interested, number 2 is cerebral cortex. Number 3 is called the basal ganglia. It's involved in dopamine. It's of great interest to people who are studying substance abuse. A lot of drugs, mood-altering drugs, target neurons in that region. And then, for example, number 4 is fiber tracts. And here are some spatially variable genes aligned to those patterns. So now I'm going to talk a little bit about trying to decompose variation into spatial and non-spatial components. So we can think about this at 2 possible angles. One would be from the gene perspective. For a given gene, how spatially variable is that gene? Can we rank genes to find the ones that are most spatially variable? Or we could also be interested in observations or spatial locations and say, for this particular location in our tissue, is the expression pattern driven more by its cell type identity or by the pattern of gene expression by its nearest neighbors? So the idea is essentially I'll not go into all the math details, but we're going to take W over W plus B. And that will range from 0 to 1 because it's not negative. If it's 1, then all the weight went onto the spatial component. If it's 0, it went to the non-spatial component. And similarly for each location. So once we have these scores, we can plot the histogram of the scores and kind of examine the distribution. It's very interesting that certain genes, this is just one of the data sets, we find that some of the genes are entirely determined by the spatial pattern of variation, whereas other genes have no spatial information whatsoever. So you could imagine interrogating these subsets of genes further to see particularly where in space they're varying the most and things like that. And similarly for the observations, many of the observations were strongly influenced by spatial patterns. However, there were some that were less influenced by spatial factors. So now I'm going to give a little more of a notion of the types of inference strategies that we did to fit these models, which is actually the bulk of the time and effort for the project, as I'm sure many of you are aware. So in terms of fitting models that involve Gaussian processes, even a simple univariate model with a conjugate Gaussian likelihood with a closed form solution will require cubic time and quadratic memory in terms of the number of spatial locations. It's a well-known limitation of Gaussian processes. When you think about cubic complexity with tens of thousands of observations, it's just not going to work. We're interested in non-Gaussian likelihoods, non-conjugacy. So that's also a limitation or a difficulty to overcome. And from a numeric standpoint, we got to do a lot of Cholesky decompositions because we have all these integrals that are involving multivariate Gaussians. And usually we need to invert some matrix or compute some log determinant of a giant covariance matrix. So dealing with the numerical instability in the Cholesky decomposition is another challenge. So two prominent approaches that we are interested in from this computer science and machine learning world: variational inference using inducing points, and from statistics, nearest neighbor approximations, which is actually more for future work. How am I doing on time, by the way?
It's 4:15 now, so probably another 5 minutes or maybe a little bit more.
Okay, sure. So I'm going to kind of gloss over some of this because we're running out of time. But the basic idea of Bayesian inference more broadly is we combine a likelihood and a prior. So here the likelihood would be, say, a point Poisson count likelihood. The prior would be some sort of Gaussian process. And then we want to infer the posterior. But in order to do so, we have to compute this intractable integral. And the strategy that we adopted is to use variational, whoops, this is in the way, variational inference, where we constrain a family of probability distributions that is tractable, and then we try to find the best representative of that family using an optimization procedure that minimizes some kind of divergence, to the true posterior. And that divergence is used to Kullback-Leibler as the metric, or as the divergence. Whoops, I think I've lost control here. So an example of that would be approximating a skewed multivariate Gaussian with a family of isotropic Gaussians and centering that in the right place. I think I'm just going to skip over this, but this is sort of the details of how we actually specify an objective function. And it turns out it's a lower bound on the evidence, known as the evidence lower bound, or ELBO. And in fact, the well-known expectation maximization algorithm is a special case of this procedure, where the bound becomes tight. In our case, the bound never becomes tight because we're constraining the family to be tractable. And that doesn't include the true posterior. So the strategy of inducing points is very similar to using regression splines. You have a set of knots that's much less than the number of actual observations, and the complexity becomes cubic in the number of knots as opposed to cubic in the number of observations.
So how many inducing points are you using for each of them?
Right. So for the 30,000 data, we were able to get it up to 3,000, and that was sufficient to produce those visualizations. Of course, for larger numbers of observations, you'd like to have more inducing points to maintain that accuracy, but you do hit a computational wall because it's still cubic complexity in terms of the number of knots.
So 3,000 seems reasonably large.
Yes, it seems like a lot, but with a lot of fine scale spatial complexity, you will start to miss things if you don't have enough knots. So here's how we kind of initialize these inducing points by just doing a k-means clustering on the spatial domain to make them sort of equally spaced across an irregular. We were not able to use like just a regular grid. So sorry, I'm just moving quickly in the interest of time. We can come back to this if people are interested. I think I'll skip over some of these implementation details and just quickly mention a couple of future interests. Yeah, in order to overcome this limitation with the inducing point knots, I'm very interested in taking ideas, combining the variational inference strategy for non-conjugacy with the nearest neighbor Gaussian process, which is a well-known technique from spatial statistics literature. And just to mention that a lot of these techniques could just very easily be applied to high-dimensional environmental data with forestry or other environmental applications. We're very interested in exploring defining Gaussian processes on manifolds as opposed to on Euclidean domains. For example, in this embryonic tissue, you can see there's big gaps between the layers of embryonic tissue, and defining nearest neighbors or spatial correlation according to Euclidean distance is likely to be highly misleading because the cell types are arranged in these layers. So I think taking an approach like a Gaussian process defined on a nearest neighbor graph, which is what we're showing here, this is somebody else's work. But I think taking that idea and applying it to spatial transcriptomics with a manifold structure could be very promising future direction. And I've mentioned this before, but I think there could be some overlap in the future, maybe more distant future, with taking some of these ideas, techniques from spatial literature and extending them to the temporal domain, with applications such as human activity recognition with wearable devices. I think I'll skip over the other projects in the interest of time. Thank you to my collaborators, and be happy to take any questions.
Questions, I guess, first here, and then we can go online. Questions here?
Yeah, so I have a question. So the graph you show is quite impressive, the factors. So you show the clear the brain structures. And so have you looked at and compared with this type of approaches with those the spatial domain detection type of approaches where they are sort of spatialized to detect the spatial domains? And are they able to detect a similar set of spatial domains as your method, or do they detect a slightly different set of those data?
I'm not sure. I'm not very familiar with those methods.
Okay.
Maybe you could send me a reference and I could try it out.
Yeah.
But I
Those are like the SPAGCN, the base space.
Oh, that one
those
I've heard of. Yeah, that would be worth considering. Certainly, there's many possible ways to segment spatial data. I think the main advantage of our approach is we have this sort of integrated model that does everything. So, but I will concede that sometimes doing everything is not necessarily doing everything well. So for very focused tasks, it's possible that a more focused approach could be advantageous.
I have a question, but let's just, we'll go on. Is there anyone on Zoom who has a question other than?
I do, Walter. This is Veera. Can you hear me?
Go ahead, Veera. Yep.
Yeah. So, Will, great talk. I think it was very clearly explained and very nice, cool method. I had a couple of questions, one more technical, one more application oriented. So the technical question, when you factorize the non-negative spatial factorization, do you let each of the factors have their own spatial correlation pattern, or are they shared across all factors? So I guess the Mettern parameter or the Mettern kernel that you imposed. So that was one question. The other question was, how would you account for, like, or how do you or do you account for over dispersion? You mentioned like an excess of zeros, right? And how does that come into the models now, specifically for the spatial single cell data?
Yes, thank you for those questions. So to answer the first question, we do allow a separate length scale and a separate amplitude parameter for each component. And we also allow a separate parameter for each of the spatial input dimensions. So it's not isotropic, it's not shared across components. And we found that the isotropic versus anisotropic was less important for these particular data. The pattern of spatial variation was not highly skewed across one, with a long length scale on one axis and a short length scale on another axis, as it might be the case with some environmental data, possibly. But I think it's very important to allow each latent component to have a different set of kernel hyperparameters, because you want to allow those functions to have very different patterns across space. And if you constrain them to all have the same length scale, for example, a pattern with a very broad, spatial variation would then be trying to encourage another pattern to have its same sort of range of variation. So I think that assumption would be a bit inappropriate for this type of data. And it's not that hard to have a different parameter for each, because we're doing it all through TensorFlow with automatic differentiation. So just as a matter of defining a different shape of tensor. And there's a little bit of slowdown in computation due to a large number of parameters, but it's only scaling linearly with the number of components. So that's not a huge concern.
And by that, you mean the number of factors, right? It's a low dimensional representation. So yeah.
Exactly. Yeah, I'm using the term components, to refer to the number of the dimensionality of the factors. Yeah.
Got it.
So then to answer your other question, I think I forgot what it was. I'm sorry.
Sorry, the over dispersion.
Oh, over dispersion. Oh yeah. So I've been showing examples where I use the Poisson likelihood. Our implementation is completely modular. We actually did do some experiments with the negative binomial likelihood, where we allow every feature to have its own dispersion parameter. And we didn't see any dramatic change in the goodness of fit on our held out prediction task. Of course, that's not the only metric that matters, but we just chose to go with the Poisson just to keep it simple, so we didn't have so many possible things to show in our comparisons. But yeah, I think using a negative binomial or some other over dispersed count likelihood would be perfectly appropriate for these types of data.
Other questions online?
Hi, this is Jian. I have a follow-up question with Vera. So you mentioned that you actually have a hyperparameter for each component, right? So and you assign a different hyperparameter or same hyperparameter, and how actually do you think that is identifiable from your model?
So you're asking whether I put a prior on the distribution of those kernel parameters?
Yes.
No, I did not. So okay, I can tell you that. First, let me say I did not put a prior on those parameters. I only put prior on the latent variables, the latent factors. The actual parameters of the kernel are simple point estimates. So it's often referred to as type 2 maximum likelihood in the machine learning literature. It's a kind of variational expectation maximization procedure. We're doing the expectation with respect to the latent factors and maximization with respect to the loadings matrix, which is just a point estimate. and the kernel hyperparameters. In terms of identifiability, this is part of the reason why we chose not to learn the smoothness parameter of the kernel. There's been some theoretical work that I was made aware of by my fellow postdoc Didong Li. That shows that essentially the combination of amplitude, length scale, and smoothness cannot be identified. You can only identify 2 out of the 3. So that's why I chose to fix the smoothness parameter, because that's actually the hardest one to optimize, and try to learn the other 2.
But how did you fit those parameters? Smoothness parameters?
Right, I just initialized them randomly. I think I initialized them to some sort of arbitrary value that doesn't lead to failures of the Cholesky decomposition. And then, the way that the implementation works in TensorFlow is you sort of register them as differentiable variables. And then the optimizer will collect all of the differentiable variables together and compute gradients and apply those gradients automatically using momentum and so forth to learn them.
So you mean you still update smoothness parameter or not, or you basically just fixed it?
Oh yeah, we update it. You have to update it. It's hard to know what the right value is a priori. So we're just
basically you
learning.
still estimated.
Exactly, yeah, with maximum likelihood, yeah.
Okay, so you basically update all the kernel parameters, not just the fixed smoothness parameters.
We learn all of them except the smoothness parameter.
So smoothness parameter is fixed, basically.
Exactly.
Right?
Yes.
Okay, so then, but I still don't understand how you pick a smoothness parameter.
Oh, how did I pick it? Oh yeah, so that's sort of I have to go back to that slide. I don't know if you remember, there was a slide with realizations of the Gaussian process, and some of them were very smooth, and some of them were more jaggedy. So basically, we just said that we want functions that have the ability to learn some, not discontinuities, but some non-smooth boundaries between tissues. We want something that's not extremely rough, but not extremely smooth either. So it was a fairly arbitrary choice.
Okay.
We knew that the squared exponential was too smooth. So I experimented around with the Matern 5/2 and the Matern 1/2, and really I don't think it's that big of a deal. But that could be a good thing for future work.
Okay.
Thinking about cross-validation to choose the right smoothness parameter or something like that.
Okay, that's good. Thank you.
Other questions online?
Walter, I have a question, or maybe a comment, rather. Yeah.
Go ahead, Peter.
Yeah, the Peter Song, and I think I really appreciate the sort of Bayesian machinery and methodology. It has great power to solve a lot of problems. But for particular data you're looking at, I think that the frequentist method, it will probably offer better, a more sort of the justified approach, because you're looking at the counts, the spatially correlated count data, high dimensional. And I think this kind of things will challenge the Bayesian method a lot. And you want to do this Klossky decomposition with the negative coefficient, right? I would say that if you go cupola model, where you can explicitly construct the spatial covariance structure, and then you can use composite likelihood to do the estimation. As we show that this composite likelihood, very similar to the idea of the nearest neighborhood approximation. You look at the low dimensional likelihood objects to do optimization. You can even estimate the smoothing parameter, the Mantic class. That has been showed in the JRCSP paper we published about 8 years ago. So I think that if you formulate problems slightly different using this composite likelihood approach, I think you have a lot of more stable numerical result rather than doing a lot of this random sort of initial values. That could be a lot of more guided solution, faster solution because you don't need to really evaluate the solence dimension in the likelihood computation. And the Slosky composition is essentially a lower rank projection. You really need to work on the covariance matrix for the projection to lower rank latent factors. I think in this particular context, I think the frequentist solution, using the Copula model and the composite likelihood will give a lot more stable, faster solution to your problem. And I don't know if you are aware of this sort of frequentist sort of methodologies I mentioned. I know that you're primarily working in the field of Bayesian statistics, but I think there's another set of the sort of tools that can probably offer better solution. So that's my comment. Maybe I don't know if you have any comments on that.
Oh, just thank you for that suggestion. I'm vaguely aware of Copula approaches, but certainly not my expertise. But I'll definitely check that out. You said JRSSB?
Yeah, JRSSB paper, and also Jen and I work on another paper on the Copula published by Metrics as well, where we work on the composite likelihood. Basically, we project this high dimensional likelihood to lower dimensional likelihood, so that you can estimate the loading coefficient very easily. Because in the Copula setting, that you can explicitly specify the covariance structure in which you can incorporate some like Maton class, for example, or a Gaussian process. Either way, you want to model your spatial sort of process as part of the formation of your covariance structure, and then then you can estimate the whatever the low dimensional sort of structure of your covariance structure, right, using composite likelihood, which is like two-dimensional likelihood.
Oh, that's very nice.
It's very fast, and you don't need to really worry about 1,000-dimensional likelihood. I mean, this updating is very, very sophisticated, unstable. You have to try different prior hyperparameters and massage a lot of things in the process in order to get some reasonable results. And I think that composite likelihood or composite likelihood that can give you a lot more stable numerical performance. We are interested looking at some estimation related covariance structure. Yeah.
Thank you. Yeah, it's a great suggestion.
Yeah.
This question is really quick, and I think it's not too challenging, but just to complete your work. So if someone wants to, I want to infer cell type rather than just doing these continuous factors, would you advise to use those factors to get to the cell type information?
Right, that's really a challenge, and we haven't solved that problem. I actually had a bullet point for it in one of the slides I skipped over. But I don't know if you're familiar, there was a paper from Dylan Cable et al., where they had this notion of first identifying cell types and then looking at spatial variation within a cell type, which I think is very interesting. So the idea with our framework would probably be we need to build in some sort of covariates, or we know the we know the cell types through some kind of marker gene approach or something.
I thought it was a straightforward if you use those vectors and try to use some standard clustering method, you can.
Well, I mean, it's true that in our paper, we tried to find what cell types were most represented in each of the factors. And in many cases, one factor did correspond to one cell type, but it's not really guaranteed by the way the model's defined. Because there's not any real discrete latent variable that would indicate a cell type. So
you can just cluster those 10 factors together into a certain cluster. That doesn't work.
Yeah, you could try it. I just don't want to promise. I don't want to make promises that I haven't verified on my benchmarking. But yeah, definitely like thinking about cell types as opposed to continuous spatial functions is important future direction of much interest to people who are actually running these assays.
All right, let's thank the speaker one more time, and Yeah. I guess online, we can clap here. All right, thanks, everybody.
So should
I will do